Hello and welcome back to Microbiology Bio120. This is the second carbon cycle lecture, and this lecture is about the parts of the carbon cycle that we did not cover in the last lecture. Specifically, this lecture is about respiration, fermentation, methanogenesis, methane oxidation, and carbon acquisition. We'll also be talking about metabolism as well. We're going to talk about what goes on inside the cell of many of the organisms, but remember to think about where on Earth each of these reactions is occurring and how it can contribute to the global carbon cycle. Here's the same view of the carbon cycle that we saw in the last lecture. Some of the things that we're going to be talking about can be traced directly onto this cycle, but some you'll have to infer based on your understanding of microorganisms. As we talk about the various parts of the carbon cycle, think about how it leads to another part of that cycle, such as how carbon fixation or autotrophy leads to respiration or heterotrophy. And always remember that microbial metabolisms have global impacts. Recall that every year we see an increase in atmospheric CO2 concentrations as microbes in the soil decompose the leaf litter that has been shed that winter. Also remember that microorganisms have two general types of metabolic reactions, and these play a role in every part of the carbon cycle and every other biogeochemical cycle on this earth. Microbes are transforming elements as part of their metabolisms, and all metabolic reactions are either anabolism or catabolism. Anabolism is just biosynthesis, or the construction of cell components, while catabolism is breaking down chemicals or using light to make energy. Let's start off with catabolism. So one way that microbes can catabolize is by using organic matter. And there's two main methods of using organic matter. One is respiration. It can be either aerobic or anaerobic. And the other main way of using organic matter as a microorganism is via fermentation. So respiration is oxidative phosphorylation. It's the reduction of an exogenous electron acceptor, and O2 or another oxidant is used as the terminal electron acceptor. With fermentation, we have substrate-level phosphorylation. It is the reduction of a compound derived from the electron donor, and there's no exogenous electron acceptor. First, let's see what's going on in fermentation, or substrate-level phosphorylation. ATP is made directly from energy-rich intermediates. With this metabolic reaction, ATP is made by the direct transfer of a phosphoryl group to ADP from another phosphorylated compound. You can see those energy-rich intermediates that are shown here. This is different from what happens during respiration, which is oxidative phosphorylation. In this metabolic reaction, ATP is produced via the proton motive force. You can see that a proton gradient is created. We'll talk more about how this happens in a bit. And this proton gradient powers the proton motive force. The proton motive force is used to power ATP synthase, which makes ATP. However, in both the case of respiration and fermentation, ATP is made. So oxidative phosphorylation and substrate-level phosphorylation are each types of catabolism. If you would like more information about substrate-level phosphorylation, this is a great website to visit. So now we'll talk a little bit more about each of the processes: aerobic respiration, anaerobic respiration, and fermentation. But we'll start with aerobic respiration because that tends to be the reaction that people are most familiar with. With aerobic respiration, the net reaction of which is circled in red at the bottom, you can see that organic matter is combined with oxygen to make CO2 and water. But what's really happening? So we can see the half reactions above that. The organic matter is oxidized to CO2, protons, and electrons. Those electrons are shuttled through electron transport chains and eventually put on the oxygen, which gets reduced to water. So oxygen serves as the terminal electron acceptor. But remember that there are many different types of redox reactions. Right now we're focused on the redox reactions that utilize organic carbon or heterotrophic processes, but there are many potential oxidants for organic matter, as shown here. You should notice that oxygen is the best or strongest oxidant as it provides the most energy. It's the furthest away from glucose, and it has the largest difference in redox potential, that is, the most negative delta G. So like I said, in respiration, an electron or electrons are taken from organic matter and shuttled through electron transport chains. Those electron transport chains are used to create the proton motive force, pump protons across a gradient, and create the proton motive force. And then the electron chain eventually donates the electron to oxygen in the case of aerobic respiration, or some other oxidant in the case of anaerobic respiration. But of course, you can get more specific in terms of the type of anaerobic respiration that is occurring. For example, if a microbe utilizes nitrate as an oxidant to oxidize organic matter, then it is performing heterotrophic denitrification. And if it's using sulfate, then it's using heterotrophic sulfate reduction. Recall that a heterotroph is just an organism that uses organic matter as a carbon source. This graphic is showing you the process that a molecule of glucose, or organic matter, goes through inside a cell to become the energy currency of cells, ATP. And so we're talking about catabolism here. It's breaking down organic matter to make energy, ATP. You may recognize this image from biochemistry or cell and molecular bio. This process is assuming that the organic molecule is glucose and the oxidant is oxygen, which is kind of an idealistic situation. It's important to have a basic understanding of how and when ATP and other products are derived, so we will go through it briefly, but just remember that there are many different types of organic matter and many different types of oxidants that are available. And so the yields and exact biochemical pathways will change depending on that. So assuming you start with glucose, glycolysis in the citric acid cycle will take place, producing ATP and CO2, but also producing NADH and FADH2, which each also holds electrons. The NADH and FADH will take electrons to an electron transport chain where the electrons will be transported from high to low energy so that that proton gradient can be created. The proton motive force then makes a lot more ATP. At the end of the electron transport chain, oxygen acts as the terminal electron acceptor and thus becomes water. So this is just a summary slide of some of the big takeaways from aerobic respiration. With aerobic respiration, remember that it's a catabolic reaction. Glucose or some other organic molecule is completely oxidized to CO2. Electrons from the oxidation of glucose, or that organic molecule, are transferred to electron carriers, NAD and FAD. But remember, glucose, or that organic matter, is the initial electron donor. NADH and FADH2 carry the electrons to membrane-bound electron carrier complexes, and then protons are pumped across the membrane to create the proton motive force. And then the proton motive force produces large amounts of ATP. Remember that this is a catabolic process, and so energy is being produced. The idea is the same for anaerobic respiration, such as using nitrate or sulfate as an oxidant instead of oxygen. It's just that less ATP will be derived because it's not as energetically favorable. Additionally, the electron transport chains will differ. So now let's talk about fermentation. Fermentation is kind of the exception for the statement all microbial metabolic reactions are redox reactions, because fermentation involves the incomplete oxidation of an organic compound. There are actually no exogenous oxidants, like oxygen or sulfate, that get reduced. However, redox reactions still happen within the cell. Fermentation only occurs under anaerobic conditions, and it is usually a last resort as very little energy is derived from the process. So if fermentation is a last resort, where does it actually occur? Well, it happens in places that are anaerobic and have been anaerobic for a long time, so all the other possible oxidants have been used up. It happens at the bottom of lakes, in the sediments, in swamps and bogs. It happens in really waterlogged soils that have been waterlogged for a while. It happens in the ocean and sea sediments at the bottom, inside humans and other animals in our guts, and in decaying fruits, animals, and plants. So if you're starting off with glucose, fermentation starts off exactly the same way as respiration. You can see that glycolysis takes place in stage 1. ATP is used and you eventually form pyruvate. During stage 2, you have some intracellular redox reactions and ATP production. Then in stage 3, more intracellular redox reactions occur and fermentation products are formed. Recall that there are no terminal electron acceptors for fermentation, so microbes are not using electron transport chains. But that's fine because they do not generate ATP via the proton motive force, so they don't have to pump protons to make a gradient. They use substrate-level phosphorylation. However, because there are no electron transport chains, the NADH that is formed by glycolysis cannot be reoxidized when the electron is pulled off by the respiratory electron transport chain. Therefore, NADH can actually accumulate in the cell, so an important step in fermentation is the regeneration of NAD+. Lactate dehydrogenase is an important enzyme that pulls electrons off of NADH and in the process also generates lactate from pyruvate. And this is why fermentation is not a true redox reaction. You can see here that the electrons that are used to reduce pyruvate to the final product were actually derived from the same compound that pyruvate was actually derived from. So the electrons came from glucose and lactate was formed from glucose, with pyruvate being made intermediately. There are no two different half reactions combining to make a redox reaction here. So lactic acid fermentation is one type of fermentation. There is also alcohol fermentation, in which alcohol dehydrogenase regenerates the NAD+, and ethanol is the final product. This is a very important form of fermentation if you enjoy certain types of beverages. So now we've discussed carbon fixation, both photoautotrophic and chemoautotrophic, in addition to heterotrophic reactions, specifically aerobic and anaerobic respiration and fermentation. We have yet to speak about reactions involving methane, so let's do that now. Methane is a very important part of the carbon cycle, and microbes are the only way methane is biologically produced and consumed. There are abiotic ways to generate and consume methane, though. Methanogenesis, or the generation of methane, is only done by certain types of archaea, while several different types of prokaryotes are able to oxidize methane, also called methanotrophy. A lot of energy is to be had by oxidizing methane with oxygen. You can see that they're quite far apart from one another on the redox tower there. But other oxidants can also be used by methanotrophs. Conversely, not much energy is produced via methanogenesis, when CO2 is used as an oxidant to oxidize organic matter. Additionally, many methanotrophs rely on the products of fermentation rather than directly oxidizing organic carbon or sugars. You should be able to understand where methane production or methanogenesis and methane oxidation or methanotrophy fit into ecosystems in the carbon cycle. Think about where you might find each of these processes, both globally and locally. This image is showing a potential methane cycle in a terrestrial system. Many soils are anaerobic at depth, and methanogens will start to resort to methanogenesis. But when the methane bubbles up to areas where other oxidants, such as oxygen or nitrate, are available, methanotrophs oxidize the methane in order to gain energy. So methanogenesis is the generation or production of methane. To reiterate, not a lot of energy is derived by combining these particular oxidants and reductants. So the organisms that carry this process out will only have a competitive advantage in locations where there is ample organic matter that is being fermented, and there are not a lot of other oxidants available. There's not going to be any oxygen, there's not going to be any nitrate, and there's not going to be any sulfate available. On land, these conditions are often found in areas where the soil is inundated or waterlogged. Methanogenesis is also found in sediments at the bottom of lakes and in the ocean as well. This figure may look similar to the CO2 curve that we've seen before in this class, but it is actually showing increasing atmospheric methane concentrations. Like CO2, methane is increasing globally, and it also experiences seasonal cycles for similar reasons to CO2. It is also a greenhouse gas, one that has an actually stronger global warming potential than CO2. And so an increase in methanogenesis by archaea and the release of methane deposits that were frozen in high-latitude lakes, soils, and sediments are of concern at the moment. It's really important to remember that a lot of the organic matter that exists in the environment does not exist as simple monomers or glucose. It exists as very complex compounds, and microorganisms are very, very small. So heterotrophic microbes and fungi will excrete extracellular enzymes that degrade the complex molecules that exist in the world. This turns them into monomers so that microbes can transport them into their cells. Organic matter in the soil is a wonderful source of electrons, and it seems odd at first glance that there would be any organic matter left in soil, given the plethora of microbes present in soils that would jump at any opportunity at an energy source. However, because the compounds are being digested extracellularly, other microorganisms can cheat and take in monomers that they did not actually digest. This creates an interesting ecological interaction in many soils, as it is less costly to cheat, but if everyone cheated, nothing would ever get degraded. A combination of cheating and the fact that some soil organic matter is so complex that no microorganisms can actually make the extracellular enzymes to degrade it, both contribute to keeping organic matter in soils. Additional factors that contribute to a lack of degradation of soil organic matter or soil organic carbon are the fact that sometimes strong oxidants are not available as well, or perhaps the temperature may not be amenable to certain microbes, or the pH may be wrong. But even though some soil organic matter remains complex and does not get degraded, a lot is able to be hydrolyzed and turned into smaller molecules. The smaller molecules are transferred across the cell envelope via active transport systems that are usually molecule specific and require energy input. But after the molecule is inside, the cell can then harvest electrons from it. So this is a general flowchart of what happens to carbon molecules in the environment. You start with complex molecules that are then turned into monomers via extracellular enzymes. The monomers can be taken up by microbes and converted into intermediate compounds like pyruvate via glycolysis. The intermediates are then turned into their final product and excreted from the cell as waste. You should understand by now that organic molecules are very energy rich. They have a lot of electrons. They are the reductant of choice for many microbes because of how much they like to donate electrons. The bonds in the organic molecules hold the electrons, and once monomers are inside the cell, the bonds can be cleaved and the energy can be harvested. Here are some delta G values for different types of bonds that could be found in monomers. Remember that organic molecules are molecules that have direct carbon-hydrogen bonds, but they can have other compounds as well in them, such as amine or carbonyl groups. Generally speaking, the larger the compound, the more energy it contains, just because there are more bonds. So organic molecules that just have one carbon in them will have less energy than a 2-carbon molecule. The oxidation state of the molecule will also affect the energy content, with the most reduced compounds having the most energy. Microbes utilize substrates and the energy they get from those substrates for cell growth, or anabolism, and Ys is a measure of that. Ys is the amount of cells per unit of substrate used, the amount of cells that are produced per unit of substrate used. Ys varies for different bacteria. Strains may use different biochemical pathways to consume different substrates, and different pathways give different yields of ATP per mole of substrate used. So Ys reflects the relative efficiency of each strain in harvesting energy, or harvesting ATP. YATP, or the ATP yield, is a bit different from Ys, but it's very related. YATP is just the amount of cells generated per mole of ATP used, and it's a measure of biosynthesis efficiency, or anabolic efficiency. Here are some examples of Ys, here called Ymax, and YATP. You can see that YATP is really similar per mole of ATP used for any given pathway, while Ys, or Ymax here, varies a little bit more. A good rule of thumb to remember about microbes is that if there is a source of chemical energy available, some microorganism has evolved a way to degrade or use it. Where there is a will, there is a way with microbes. So now we've talked about all the processes in the carbon cycle that are pictured here, with the exception of carbon monoxide oxidation. But for that, it's just oxidation, it provides energy, and where there's a will, there's a way. So as you're studying the different aspects of the carbon cycle, really think about how each process leads to the next, and where you would find each on Earth. So I want to leave you with something that affects all metabolisms on this planet. It's called the metabolic theory of ecology. So metabolic rates are fundamentally influenced by both temperature and mass. Most chemical reactions, including those involved in metabolism, don't just happen spontaneously because there's an energetic threshold called the activation energy. As the temperature increases, there is more energy available to overcome that energetic threshold, and hence the reactions develop at an exponentially faster rate until you exceed the thermal tolerance of the enzymes and the organism involved. But also, the body mass of the organism plays a role as well. So small-bodied organisms tend to have higher mass-specific metabolic rates. And at higher temperatures, either via internal endothermy or external, such as with climate change, both lead to higher metabolic rates. So even though bigger things have a higher metabolic rate just because they're bigger, microbes actually have a higher mass-specific metabolic rate. And because there are so many microbes on this planet, this has implications for climate change and all the metabolic rates that could relate to climate change, such as those that produce CO2 or methane, both greenhouse gases. That concludes this lecture.